My guest today is Dr. Barry Goldman. Barry and I, another trip back down memory lane, were classmates and lab mates at the University of Utah and back in graduate school. And if I recall correctly, pretty wicked with a croquet mallet.
That was not the question or the comment I expected, but yes, I was at least within the top 10 of our lab in croquet.
We used to play croquet and volleyball, and there were no boundaries on the croquet field when we
Yeah,
played during our little afternoon breaks.
yes.
Um,
It was an
yeah,
enjoyable way to waste a little bit of time while you were thinking about and waiting for things to grow.
right. Right. So Barry is the founder and chief scientific officer of Pluton Biosciences. He led the microbial testing pipeline for Indigo Ag and served as the microbial discovery lead for Monsanto. He's also published over 30 peer-reviewed studies and is an inventor on more than 10 patents. Barry, welcome to CC Life Science.
Thanks, Chris.
This is going to be a fun one today. We're going to the ag field, which is a little bit different for us, and the microbiome, you could say. So we're talking about bacterial cover crops or microbial cover crops and how that can impact carbon storage. So tell us about Pluton Biosciences, where the idea came from, and what your focus is.
Okay, thanks, Chris. Yeah, and I'm thank you for inviting me on to talk about this stuff. I have a lot of passion around microbes. You and I spent a lot of time thinking about those. So the idea from Pluton really evolved over quite a bit of time. It was I started seeing that in science we had started uncovering that the number of microbes was almost unlimited. The recent sequencing and computational biology aspects of technology made us understand that there may be as many as 1 trillion species of microbes in the world, and which is roughly 1,000-fold more than anybody had even considered. And when you look at the microbial products and how they've changed humanity, just the ideas of antibiotics and cloning, and now recently CRISPR, all come from bacteria. And so the idea was, well, if this much has been done and we only understood a tiny fraction, why don't we keep looking in places where we really haven't been looking before, to start looking at those microbes that people just haven't looked at before. And so the point of Pluton was to start finding a different way to look at those organisms. And so that's really where the idea came from. And we started about 4.5 years ago or so, and started coming down with this idea about how do we look at microbes that are rare, but might provide new ways for us to solve big problems for humanity.
So you didn't necessarily have an outcome in mind. It sounds like you were
Mhm.
saying, what could we find bacteria that are capable of doing?
I think there was a little bit of an outcome. I'd worked in agriculture for about 25 years and realized there was a lot of issues. I think the biggest of the issues that I was looking at was insects becoming resistant to chemistries and to genetically modified plants that contained essentially proteins that kill the insect. It was a cool technology. It was developed in the mid 90s. And essentially, the insects that feed on our crops, this would just kill them. So the only way it would kill them is if they munch on the crops. So how's that for targeted, right? The moth comes around. It's actually the larval stage of the moth comes around, and it only dies after it chews on the plant that it wants to. And so, as far as the farmer is concerned, well, that's the one I want dead. I don't care about anything else, but the thing that eats my crop, that's the one I want it, and I want it dead before I have to do anything about it. And so, the idea of putting a protein that kills the insect, that's specific just for that insect, into the plant, and so that it kills that. That was a revolutionary idea. It started in like late 70s and early 80s, and it took about 15 or 20 years really for it to become a novel product. But now, 25, 30 years later, the insects are becoming resistant to those proteins. And so the question is, can we find new proteins? Can we find new chemistries that are safer for humans, that can protect our crops, can reduce the amount that a farmer loses, feed more people on less land so we can protect the land like rainforests to protect all of humanity? And so really that was the target, was to say, how do we find new molecules that will protect crops?
I just want to say I love that vision. Like you're just thinking bigger. Like there is a specific goal in mind, but here's what could happen if we could do this. So
Right. Yeah,
kudos
and
to you.
and I guess the phrase we had when I started raising money and I started asking people to join this little group was, and this will be common to you, is like, if you can imagine it, a microbe already does it.
I was gonna say that. I mean, like
Comes right out of our lab experience. That was what we were taught. If you can imagine it, a microbe's already doing it and doing it better than you can imagine. And
right. We
so
just
the
need to
idea
find
is
them.
you just need to find them. And it's a very different model than the last 20 years of saying humanity can create it, which is great. We've done terrific with it. But what if we've never thought of it? And there are microbes that have been doing these for, 1 billion years. Literally 1 trillion microbes have been thinking about these kinds of things, thinking anthropomorphically, thinking about this stuff for 1 trillion years, and they've solved pretty much every problem. So you think about that, as I was thinking about this whole idea, you think about the plastics that's sitting in the Pacific Ocean. Well, there are microbes that can degrade it. So when I was thinking about this, I said, we can solve anything, right? Now, to start a company, you can't try and solve everything. You've got to focus on what you know and what you can deliver. And so what we focused on was something that I knew, which was agriculture, and a space that I thought was amenable to ways for us to go after, these new microbes.
Awesome. All right. So remind my non-ag friends, which is
Yeah,
probably most people who listen to my
yeah.
podcast, what a cover crop is. Because I think I only learned this when I bought a house and was trying to figure out what to do with my garden in the winter. And explain the concept of a microbial cover crop.
Okay, so the idea of a cover crop is you have your standard crop during the season, and traditionally, what's been done is you would till and essentially take a tractor over it and put that crop back into the ground, like take the stubble from corn or the stubble from soybeans or something and put that back into the ground. But the problem is, it leaves the field accessible to too much rainfall and to wind and different kinds of erosion, and so you lose nutrients and you lose soil. So the idea of cover crops, and this is stuff that's been around for literally thousands of years, is to plant something else into that soil to protect the soil, protect it from rain, protect it from wind. And if it's the right kind of nutrient or right kind of cover crop, maybe put a little things back in. So a lot of them are nitrogen fixers. So alfalfa is a pretty good cover crop, and it puts a little bit of nitrogen back into the soil, and so it makes it a little healthier. In addition, a good cover crop will also put roots into the soil and sort of break up that structure. When tractors have gone over it, people have walked over it, the soil compacts. Rain and other things also compact the soil. And so by getting roots into there, then it can sort of break up the soil and make the soil overall healthier. The problem with it for growers, for farmers, is that it costs money. I mean, it's expensive. And so you have to, for a farmer that's sitting at the edge of how much they make and how much they can bring in to feed their families, it's like, well, that's extra money. I've also got to take my tractor afield another time, and that costs money. And then when they're done, they're weeds. You don't want those things in the field. They're pulling nutrients out after they've done their job. And so for most farmers, the cost going in is $50 to $100 to put it in, and then you have weeds, and then you've got to do all this extra work for an improved soil. As a consequence, only 5% of farmers actually use cover crops. And in some places north where the cover crop can't go, so let's say you go to North Dakota or Minnesota, well, it gets too cold too fast, you actually can't do a cover crop. So we came up with this idea of a microbial cover crop because microbes can grow in about a month and grow pretty fast. They're really cheap. That is, we come at a much lower cost than making seeds. They can still put carbon and nitrogen into the soil. They can actually, when done right, can protect the amount of rain that's coming in because you can form a crust. These microbes can form a crust. And then the farmer's done. They don't have to worry about going back out into the field to remove weeds because these aren't weeds. They're actually the same things that are found in the soil. In fact, that was the whole concept. You take things out of the soil, you find the right microbes that can do this job, and then you just put them back on the soil. And then they just get, as most microbiomes work, they just get overwhelmed by the mass of microbes that are there. And so they just go back into the soil where they belong because they're behaved in the right way.
Yeah. So as I'm thinking about that, the cover crops, as you say, become weeds at some point. Now they're competitive with what you're trying to grow.
Exactly.
And you're flipping that. Now you're boosting, you're giving some of the right microbes an advantage for a short window of time.
Mm-hmm.
And they will eventually be outcompeted by everything else that was already there. But
Right.
for some period of time, you're putting on a crust, you're protecting the soil, you're putting some carbon and nitrogen back in.
Right. And it's powered by the sunlight, right? So it isn't like they just magically do this. These are actually photosynthetic organisms, and they're just using the power of the sun exactly like plants do. And they use that power to do two things, to turn CO2 from the atmosphere into sugars, and then take N2, right, which is the basic form of nitrogen that represents 78% of the atmosphere, and turn that into ammonia. And that's a very energetically expensive reaction. And so today, we do that through a process called the Haber-Bosch process, and it uses lots and lots of energy. And in fact, it creates a lot of CO2 gases, and by mining for methane causes a lot of problems. And that's kind of that's basically the 1950s and 60s was making synthetic fertilizer from this. And it fed lots of people. We had 3 billion people in the 50s, and now we have over 8 billion people, and that's largely because of this synthetic fertilizer. But it has a bad side effect. It's impacting our climate in a very negative way. And so one of the ideas is, can we turn that around? Can we let the microbes take that energy from the sun that's just going onto the ground and turn that into ammonia the same way the Haber-Bosch process does, and feed the microbes, and then have the microbes feed the soil, and then the plants that we need to eat to grow and keep all of us healthy and happy, have them use that to turn that into productive food.
Nice. All right. Science sidebar here. I get the CO2
Yes.
and the photosynthesis part, turning it into usable carbon, sugars, etc. Where does the ammonia fit in the microbe's lifestyle in turning it to nitrogen? Just for use in biosynthesis of making amino acids? And
Yeah, absolutely. That's where it comes from. So a little more of a science sidebar. So N2 gets converted into ammonia, NH3. That is actually put onto an amino acid called glutamate to make something called glutamine, and from there it enters the cycle for all, and turned into the rest of nitrogen for the plant. So, but again, it's very energetically expensive to do that, to break those bonds apart and to put hydrogens onto there to turn N2 into ammonia. And so that evolved once in history. The idea that there are organisms that fix nitrogen. It evolved once at some magical period, 1 billion years ago or so, and some organisms figured out how to do that. Most of them, they were probably sitting at the bottom of the ocean, getting there in a place where nothing else grew. Heat's coming off the bottom of the ocean, and they were splitting nitrogen at that point through the energy generated by that heat, and adding it to make biomass. And some of the earliest organisms in the world were living there. And somehow that set of molecules that could do that went to a few other organisms. We know that best like rhizobium, the rhizobium that grows with soybeans, right? That allows soybeans to grow with less nitrogen. That same set of genes is the same basically set of genes that evolved 1 billion years ago, and now is in different kinds of microbes. But it's only found in bacteria. It's not found in fungi. It's not found in humans. It's when they say plants fix nitrogen. Plants do not fix nitrogen. Plants have microbes that live with them that allow, and those microbes provide the nitrogen for the plants.
I'm going to tell the audience, I feel like I'm coming home. We don't talk about bacteria enough on this podcast. So between you and Mike Mahan, who was a couple weeks ago
right.
on the episode, I just feel like I'm with my people now.
Right.
So you have found one or more consortia of bacteria, you're going to explain that in a moment, that can perform this function of photosynthesis and fixing nitrogen. So why a consortia and how do you isolate them?
Yeah, so a consortia means group of organisms, just any sort of group. And it turns out that some of these organisms can do both of those reactions by themselves. So they're a type of organism called cyanobacteria that can both photosynthesize and use that energy to fix nitrogen. But it turns out, and what we've seen in our lab, is they're happiest when they grow with other organisms. And so we started with an environmental sample that contained 10,000 different species. And then we grow them under conditions where we require them to make their own carbon and make their own nitrogen. That was basically it. It's a little trickier than that in the presence of light. And a few come up under those conditions. And when you say, well, what's there? We find the same set of organisms, find these cyanobacteria, but we also find some friends that always seem to come along. And that isn't surprising. As humans, we have a human microbiome, right? We have these things that live in our gut. And they evolve together. It isn't random what's in our gut. There's some very specific players that keep showing up again and again and again. And so it's not surprising that these cyanobacteria grow up with these other organisms that allow them to do their job better. And just like, cows, that there's grass and cows start eating the grass, and that's kind of the beginning of our food system. The cyanobacteria, the beginning of the food system, right? They get all the first primary, they get carbon and they get nitrogen, and they make what's called an extra polymeric matrix. So it's kind of like they excrete a lot of extra goo, is essentially what it is. It protects them, it keeps them from drying out, but other organisms go, oh, this is food for me. This is lots of sugars and all sorts of goodies. But what's happened over evolutionary time is these other organisms have provided value to the cyanobacteria. And so not maybe classically symbionts, but certainly friends. And as a consequence, when you just isolate a cyanobacteria from nature, usually you'll find 10 to 20 organisms that live within this matrix. And so we suspect what we're finding is just things that have come along and live in this matrix and provide value to the cyanobacteria and allow them to grow together. And so we just thought we would just sort of follow along what nature's already provided and make groups of organisms that can work together to solve the problem. What we find is when we do growth curves and ask for them to grow, they always seem to do better with more than one organism. So we isolate the cyano to a single organism, and then we say, well, how well does this grow compared to hanging out with its friends? And with its friends, it always grows better. And so we say, well, don't break up a good team. Have them hang out together and do their job.
All right. So as a group, how do you see them behaving in the lab? Of course, as you say, you find an isolate or a group of bacteria that with just light and minimal nutrients, no nitrogen, no carbon, make it all happen. How do you, I guess, measure their output? I guess what I'm asking is how do you measure the carbon production and nitrogen fixation?
Yeah, so there's several ways you do that. One of them is you can just grow them. If you're not supplying carbon or nitrogen, if you just separate them, if you just separate, anything and put it there, nothing will grow, right? So you can turn off lights and see how fast things grow. Turn on lights and you can just measure and weigh biomass. That's one way. That's a simple way. Another way is what's called combustion. So you literally take the biomass and you burn it. It turns into a gas, and you can actually measure the what's called carbon, organic carbon, from that. In fact, farmers all do that to say, how healthy is my field? They'll take soil samples from around their farm, and they'll send it off to say, what's the percentage of carbon in my field? And they'll do what's called a combustion assay with it. They'll send it off someplace, burn it, measure the carbon and nitrogen in their field, and they'll say, okay, looks like I'm a little low on nitrogen. I'll have to add a little more fertilizer. Or they'll say, oh, my organic matter, my carbon, is low. I'll need to do something to improve the overall organic matter, which is when they might put in a cover crop. They might say, oh, we need to increase that carbon in the field. So they do that as a way to look at the health. And we do the same thing, whether growing in liquid or growing on solid plate. So we use an agarose plate, just like agar, but no other nutrients in it. And then we're also putting it on top of soil, where we can essentially take the whole soil sample and no other inputs are there, and we combust it. And then we say, did you get more carbon when you're done, or do you have less carbon? So you measure, let's say you measure 100 grams of soil and you put them in 2 containers. On one you put your test, and when you don't put a test. And then you say, is there more carbon when we're done? And we see, yes, there is, right? And so that's how we do it, is we actually measure that. The other way is a lot more sophisticated, where you use an isotope of carbon called 13C. So it's just a little bit different weight. And there are machines that can actually measure that different. It's not radioactive, it's just a different weight. It's like having 2 lead weights, and one would be slightly denser than the other because you put something else in it. And you just say, hey, this one's different. You can actually measure the uptake of carbon dioxide, because you can provide carbon dioxide that will have this 13C in it and measure how much is taken up by the plant. You can also do that with nitrogen using 15N, slightly different isotope of nitrogen, and measure that, send it off, and do that. And we're in the process now of testing those experiments. And the reason we're doing that is measuring this stuff in the field. So you can measure it in the greenhouse and say, this stuff is great. And then, hey, farmer, believe me, I've done something in my little greenhouse. And they're great, but I live in the real world. Can you show me what's going on in the real world? And so what we're trying to do is do these isotopic studies first in the greenhouse to make sure we get it right, and then take it out to the field to see how much is getting sequestered, fixed, and taken from the atmosphere and put into the soils.
Yeah. All right. So, in addition to fixing nitrogen and adding ammonia to the soil, describe the potential benefits for the climate of doing this sort of thing.
Yeah. So this starts getting this is going to start getting some pretty high-powered math here. So, we believe we can take somewhere between half a ton and a ton of CO2 per year per application. Right. So let's
application
acre.
Yeah.
Per acre. Sorry. So I'll just make it simple, even though those numbers may not be entirely right, but I just want to keep the math simple. So let's assume it's 1 ton per acre per application per year. Okay. If you can put this, and let's say we can get the cost really low. Let's say we can get it down to like $20 an application, right? So $20 an application. I'd probably do 10, even though I know that's way too low. $20 an application. So if you can get this now on 100 million acres, you removed 100 million tons of CO2 per year. Okay. There's roughly 7 to 8 billion acres of accessible farmland. So now let's say you could do 1 ton. We're just going to say we have the entire market. We know that's not true, but let's say you have the entire market. You could pull out 8 gigatons of CO2 out of the atmosphere. Humans are putting 30 gigatons into the atmosphere. Right? So it's not that much, right? So you can now take that's close to 25%, right? 8 over 4, 15, yeah, something in that ballpark. 8 over 30, whatever that is. It's a little over 25% of the CO2 that humans are putting into the atmosphere every year. That can make a huge dent on our impact on the planet, right? And that's just one solution, right? We absolutely believe that many solutions have to be used together. In addition, we also believe we can put on 30 tons of nitrogen, or 30 pounds of nitrogen per acre. So now if you're on that for that much, you're essentially almost turning the amount of the Haber-Bosch process to zero. That's roughly 20% of the greenhouse gases that are being emitted. You put those two together, now you have a massive impact on climate change. So here's where the math gets to, right? So if let's say it's $20 an acre, 8 billion acres. Okay, the math gets pretty good. That's $160 billion, which is a lot of money, right? But imagine you could address climate change for $160 billion, which is roughly a fourth of our defense budget. That you could now start addressing climate change for a fourth of the U.S. defense budget. That's not even the world defense budget.
Right.
I mean, now you can start tackling this and give ourselves, let's say, more time to come up with even better solutions. But you can do this relatively easily. We can grow our microbes anywhere, right? All we need is a pond. We just need a place to grow them, and then we can take them out into the field. And again, we're not selling these yet, but we're in the process of trying to get these to market. Right now, we're in the process of testing in the field.
Yeah. So talk about testing in the field, that those challenges, because a farmer gives up something to have you do that, and then also the regulatory path, because I mean these are completely natural organisms.
Right. Yeah. So let me talk about the regulatory path first, and then we'll talk about farmers and the challenges they have associated with this. So the regulatory path, the regulatory agency is the USDA, and so they have a group of organisms that they say you can plant these anywhere. You can put these anywhere. Those are called non-jurisdiction. And then they have a group that say, well, you can't put these anywhere. Each of those lists is a couple hundred. It's not very many. If there's a trillion species, you're kind of outside the list, and that's what we find. Most of the stuff we have is outside the list, and it's just because it's not known. And so what we have to show is the presence of these organisms that basically came from where we said it did, and it's found in a lot of different places. So as a consequence, we have to do a lot of sequencing and identifying or finding other groups that are sequencing to say, is our organism, is it pretty much everywhere or is it pretty rare? And if it's pretty rare, then we have a tough time taking it to the field. If it's found in a lot of places, it's much easier. We can take it out and then we can do that. And so what we're looking for is a group of organisms where most of that work really well, but everybody, the USDA knows they're out there, and then we can get approval for them. So that's kind of the path that we take. Again, they are not genetically modified, so you don't have to take them through the EPA to get them approved through that path. And so you can just make sure that they can, just, hey, we know what these are. We know the sequence, we know the information, and so we're comfortable with you putting them out there and testing them. For the grower, it's a matter of when can you get these out in the field. And so we've been thinking really hard about a farmer has to take a tractor or a field a certain number of times per year, right, to do what they need to do. Can we figure out a time for the microbes to go out there that's entirely when the grower is going out there as well, so that we don't add an extra pass? And so so this extra, so it is $20, right? So that's a cost. That's a cost, and we can't get around that. Of course, that's the model is what we'd be paid on. But if we can make the cost so it's only $20, and they say, well, it's $20, but if you really do put 30 pounds of nitrogen on the soil, and it costs me, let's say, 50 cents to do that, well, that's about 15 of my dollars right there that I've saved, right? And then, so basically, maybe it's a wash a little bit in terms of that. I'm going to put on 30 pounds less nitrogen at a different time, and this may be better nitrogen. That's one of the other questions. Synthetic nitrogen, it's believed as much as 70% is lost to the atmosphere and converted into N2O, which is a really nasty greenhouse gas. And so the idea then becomes, can we put on an equivalent amount or more than equivalent amount of nitrogen, so the grower gets value on one. The second is what's a carbon market, and boy, I wish the carbon market was farther along. Every year they say it's going to be better, but it hasn't been better yet. Where there's an actual carbon market, but there's ideas around carbon market, and right now that's sitting at about $15 to $30 of ton of carbon sequestered per acre. So if they can also get value from that, and then finally, do they get healthier soil? Do they improve yields? Do they see better for the year? And so the downside is the extra effort of buying it, loading the microbes, putting it on there, making sure it works. The upside is improved nitrogen use, improved carbon for the field, and dollars in terms of savings on nitrogen and perhaps in the carbon market.
Nice. So that pretty much covers sort of the business model for how this gets paid for.
Right.
And sort of I guess we haven't really talked about is the risk a farmer takes just for testing this, like, and sort of the big picture effort that we have to all be willing to sort of undertake to make things like this go forward, right? I mean,
Yeah, and that's why, we're not going to be selling this at least for, a few more years, because we do have to show safety and efficacy, right? You got to show it does what you think it does. The greenhouse is lovely. As somebody who's been in the agricultural field for 25 years, the greenhouse is, oh, that's a nice experiment. And the field will totally change everything you know about what's happening, right? So you have to get it out to the field. You got to show it does what it's supposed to do. It doesn't have a negative impact on there. And so you take this out to fields that we pay for. We pay for the research. We hire consultants that can test it for us, know what's being applied. Usually there are groups through the universities that do this kind of work. And we'll say, here's what happened. They say, what kind of experiment do you want? They'll work with you and say, well, this is how to do this the right way if you're going to be on a farm. And we'll take data. We also have drones that fly over the fields to make sure it's doing what we want. So the first thing we ask is, well, they should be green. Do we see reflectance? They should be growing. Is that reflectance improving over time? How much of the field is being taken care of? And then you go to the field and you'll take samples, do the combustion analysis, but you'll also take samples from the top and say, are our microbes still out there? That is, we'll sequence those organisms and say, this is what we put on there. Is that what's here? And so we do those kinds of analysis as well. And we'll do that on a lot of different fields before we move to the next step. And so normally how it's done, you start with just a few fields. Then you say, hey, that worked. Let's add, maybe 10x more fields. That worked. Let's add 10 or 20x more fields. And then, while you're doing that, you're figuring out how do you grow a lot of the organisms as cheaply as you can. So, but, that's just business model. Reduce costs, increase efficiencies.
yeah. When you mention reflectance, that's impressive. If you're talking about growing enough of this stuff that a drone can see a change in the reflectance on the soil.
Yeah. Well, you can actually see this stuff, so when you go to the field, you can actually see these things growing out there. And in the greenhouses, it's really, really easy to see that stuff. And we develop reflectance, and in fact, we're working with the Danforth Center, which is in St. Louis, just not far from us. And they have something called a crop reporter. So they have a machine they call a crop reporter, which is how healthy is this plant using reflectance and all these things. And we just take our stuff down there and use it exactly the same way. So we're building those models about how to understand it in the same way they've used to look at plants. And remember, the cyanobacteria are related to the chloroplasts you find in plants, right? They're both derived from the same precursor organisms. And so the photosynthetic machinery is in fact almost exactly the same, right? So I like to explain this to people. I say, don't think of them as bacteria. Think of them as really tiny plants that are doing exactly what we want the plants to do.
That's nice. It's also, I'll just point out that this is sort of a callback to one of the very first episodes I did on this podcast, which was about machine vision and using either satellite or other airborne imagery to assess the health of plants.
Yeah, that's exactly right. And we've developed that machine vision for our lab work, right? So we do a lot of machine vision in the lab. One of our first assays we developed, so, you can grow these in liquid, great, but the farm's not liquid, right? And so you had to come up with a model for soil. And at first it was hard for us to distinguish microbes from soil. And so what our scientists did was to essentially take agarose in little 12-well wells, plates, and essentially grow it on there, starting with just a microliter material. And of course, it starts growing out using photosynthesis. And you just measure that over time. And that essentially goes from nothing to keep growing out. And you can generate, using machine vision, you can actually generate a growth rate for how fast it's growing. Allows you to compare different consortia to say which ones are growing faster, allows you to see what the reflectance is and start using machine vision to say, well, which ones work better. And pretty much we always find that the ones that grow better in our, basically surrogate soil grow better on regular soil. We see that every time. We've seen that every time so far. And so it allows us to very rapidly figure out which of these groups of organisms grow better. And then we can confirm that using different kinds of assays to show, are they also working better in soil? Can we bulk them up faster, et cetera?
Nice. Barry Goldman, this has been a treat to one, reconnect, and two, here. This is I think, a really cool story. I wish you a lot of success.
Well, thanks, Chris. Thanks.